are beginning, we've begun. All right, everybody, this is, trying to phrase it the right way. The challenging stuff starts now. I hate to say it that way. But this is, really a lot of what we're going to focus on for truly of the bacterial anatomy, how it evolves around things that we're looking at here. I frame a lot of the questions in what's distinctive between gram-positive and gram-negative. We won't get to that today. We might see it today, but without going into it in any great detail to start. What are the differences between prokaryotic, meaning bacterial structures, and what are the differences between eukaryotic, like our structures? Like we have flagella in both. They're hardly the same. They're not composed the same. You're going to see that fairly soon. So there's a lot. That's kind of the focal points. Endospores is a focal point. Again, the distinction of gram-positive and gram-negative is a focal point. Aspects of the DNA are a focal point. Plasmids. Those are all really important areas. And so I would dare say this chapter, half the first exam kind of comes out of that. It's probably a fair statement. So let's begin. I know we got to start school. It's like our fifth meeting. It's about time to start. I didn't see any enthusiasm here. It's like, well, you're just sitting there going, I don't like this idea. Well, it's too darn bad. Not what I would have said in another day and age. So we're going to look at the bacteria. So I'll give you the quick view with this one first, because the illustration in the other text. You'll see this. This is Nestor's Chapter 3 PowerPoint. And I like the illustrations in some ways a little bit better, not all of them. But this is an electron micrograph of bacteria. All this central portion here is genetic material. Okay, so there's an abundance of that. As I mentioned before, it's squished into a very, very small space, an immense amount of genetic material. So much so that when a bacteria dies and the genetic material blows out, okay, it could wrap around that cell 1,000 times. That gives you an idea of how much genetic material. It's not like an exaggeration. It's what it is. Scientifically, when you measure the angstroms and things like that. So you have, at the very least, a membrane, a cell wall, some type of exterior layer, either a capsule or what we call a slime layer. Typically, you have a flagellum. Sometimes you have these little appendages. They call them pili. Actually, they're fimbriae, as you will see is a better word. And that's about it. You have cytoplasm, and if there's an organelle inside, it's mostly ribosomes. So it's basically you have genetic material, and it makes proteins by way of ribosomes. This was really involved. There's other stuff, as we'll get to. So, I mean, it's a simplistic, but sometimes simple is better, particularly when you're learning about this material at first. So every so often I'll flip to these illustrations. That's kind of why I put them in there. So this is the textbook PowerPoint sequence that we're using here. So what do they have? They all have, all living things have a cytoplasmic membrane. It's a gatekeeper. Things go in and things go out. The really important idea of this membrane, it's not separate and distinct. Down the road, you're going to see, and all good micro textbooks call it a cell envelope because they all have a cell wall. The membrane and the cell wall are not separate, even though they are separate units. Functionally, they are not. They're married together. If the membrane is destructed, the cell dies. If the wall is destroyed, the cell dies. Okay, so envelope is a good term because you think of, to me, that means they're a unit. And when you damage the unit, you tear apart a letters, we used to see those things, mailbox. Just shut up, nobody cares. Thinks you're being funny. I still pay 2 bills by mail. A water bill and a car payment because the bank wants the car. You have to open an account. No thanks. I already have enough bank. Moving on. So cytoplasm, pretty generic, like ours. Nothing special about it. Ribosomes, those 70S, nothing membrane bound because there are none. A cytoskeleton, very limited. Maybe there's a few actin-like filaments, non-specific proteins. Maybe to some extent they act as like little underpinnings. If this is, it's like those little plastic or metal things that hold up the drop ceiling, that kind of stuff. There's not a lot of heavy-duty organization. Again, we used to think there was none. Now, in the age of advanced microscopy and molecular biology, we know. And yes, there's always at least one major chromosome. We of course call it the bacterial chromosome. That's that big supercoiled one. And yes, most bacteria have plasmids. I commented on those before with genetic engineering. Small, relatively genetically speaking, 30, 50, 70, maybe at most, genetic locations on a circle of DNA. And interestingly, by definition, that's considered non-essential. Essential is an important word in a science class. For something to be essential means you can't survive without it. Okay, so they are useful but non-essential. The information on those genetic markers are helpful. They can allow them to exist in difficult environments or produce things that may be advantageous. But if the environment would go back to less problematic or back to quote normal, plasmids can disappear and reappear when needed. So they're it's an interesting sort of an oddball thing. So, and most bacteria have at least one and frequently more. And they're tremendously important when we do our unit on bacterial genetics, which is in the second test unit. That's there. So they all possess a cell wall. The question is, how organized is it? As you will see when we get to some sort of unique bacteria, the cell wall is subject to change based on environmental conditions. It's not, when you and I think of a wall, we think of a wall. They're not all like that. Most are like that. And nearly all the cells have some type of shell that is composed of sugars that called glycoproteins. Because proteins are in the membrane, you have little short chain sugars. Our cells have. All our cells basically have their immunological markers that are there, these short chain sugars, which means why your blood type is A or B or AB or O. It's also to do with those sugars. But in bacteria, they pretty much all have them. And depending on how organized they are, they can either be relatively short or poorly organized and more or less just be like sugar on the outside. By the way, I love candy analogies. It's just amazing. It's like when you get one of those candies that have like a lot of like the sugar on the outside and heat and humidity, they get sticky. You know what I'm talking about? Like the Sour Patch Kids, if you leave them in the car, sticky. Not I would know about this. As opposed to something that's firm, like you think of a peanut or a plain M&M that have a hard shell. That's more of like something we call a capsule. So you're going to see the distinction between slime layers and capsules. I love candy analogy. It's important. I told you guys this before. See, I taught some of you folks during the fall, which is Halloween season. Okay? So it's like I had to be very cautious with my first grandchild. When she went out trick-or-treating, I had to make sure that things like Reese's Pieces and M&Ms were not tainted. Little Twix bars. So when I saw anything that looked suspicious, which is all of them, I ate them. Where are the Hershey Kisses they gave them? They look questionable. I couldn't risk it. Just saying. See, Twix bars, very much like that. Moving on. Bacteria have to have a means to get around. Most of the time it's flagella. Not all bacteria are motile. That's the term we use for mobility. You're actually likely in the lab going to test for that. Either how it grows on the plate or you're going to inoculate a tube that has some type of gel material in it, and you'll see whether or not it impacts that and spreads around. Those are measures of motility. Those that do move generally have move by way of flagella. Bacteria are, a typical bacteria is about 10% of the size of a red blood cell. They're really tiny. Okay? So the flagella, the long tail of that, is not at all anywhere near as large nor composed of the same material as the flagella in the male and sperm cell, okay, which is considerably longer and easy to see microscopically. They don't work the same way. So even though we use the term, they're not equivalent. In eukaryotic cells, like a sperm cell, like one of our cells, a flagella is called an internal protein appendage. I always keep one of these handy for demonstration. With a dog. Well, and he's willing to fill it indirectly. 75 pounds of dog. Big Phil. And so it's like the protein is my finger, the flagella, and the membrane is the bag, which is totally different, okay, than in a bacteria, they are external appendages, like a hair that's growing out. Okay? And they're different, and they're much, much smaller. Flagella in our cells are made out of, you might remember, microtubules, something called the 9+2 arrangement. Is that ringing a bell? 9 doublets with 2 little circles in the middle of microtubules. It's very important evolutionarily. But and these flagella are made out of a repeat unit of protein that's somewhat linear. And they didn't have a really great, clever name for it, so they called it flagella. Flagella are made out of flagellin. Pili, which are a shorter version of flagella, not quite constructed the same, are made out of another protein called pilin. And then fimbriae are smaller still. They're like little bristles. So if you like in the fall when you go walking in the woods, you have a sweater, and you get one of those like little burr things that sticks to it, and kind of get home and go, what's that? You've had that. Those are fimbriae. Fimbriae are how bacteria typically attach to our cells on the inside. So these are all significant proteins. And again, none of them are encased in membrane. So happy I had 2 this morning. He only used 1. So they all have an outer membrane is not the same as a cell membrane. When we go back here, they all have to have a cytoplasmic membrane. That's a standard phospholipid bilayer that we are familiar with our cells. However, an outer membrane, as you're going to see, is unique. It's something, and when I use the term unique, just like I used it with peptidoglycan, unique means you don't find it anywhere else. The outer membrane of the gram-negative cell, which we're going to spend a lot of time with on Monday, is made of something called a lipopolysaccharide layer. It looks like a typical phospholipid that you learned about in A and P1, with the tails and the heads. It's not. It's so unique, it's totally foreign to you and I. So much so that the lipid component, the fatty component of that, is always toxic to us, meaning our immune system will respond, and it will respond vigorously if it gets out. Think of a burst appendix. Okay, when an appendix bursts, it floods the interior of the abdominal pelvic cavity with basically bacteria, along with a bunch of other stuff. And the bacteria are mostly in your gut, in your colon, gram-negative. And that's why they get a thing called peritonitis, which is an immediately life-threatening situation. Like if you burst an appendix, normally it's a ticket to intensive care. Normally, not always. I had an interesting case where it wasn't when I was a resident. So the outer membrane is not the same as the cell membrane. Gram-negatives have an inner and an outer. Nanowires and nanotubes are relatively recent discoveries of structures that play a role, like say with flagella, okay, with positioning and movement. So they're not a big deal for us. The plasmid, again, is an extra little circle of DNA, pivotal role with many bacteria in how they transfer genetic material from one to another. Bacteria are very interesting. because they can transfer a useful trait from one bacteria to all the bacteria in a population literally overnight. And we see this. We have had people where they have a staphylococcal infection with one MRSA involved in that, and all of the bacteria become MRSA overnight. That's because when they can pass that genetic information in a very, very rapid period of time. We will go over that. It's called conjugation. You'll be seeing an illustration of kind of what that looks like shortly. Endospores. Talked about that ad nauseam. Inclusions first, then endospores. Inclusions are something that you'll see in the lab. A lot of this has sort of dovetails into lab. I guess the best way to explain what our lab is about, our lab is basically what we would call a bacteria lab. It means bacteriology. You're studying bacteria. And that's great, because that's really what you need to study. As much we don't have the capacity for viruses directly studying them, because it requires a whole more safety and a lot of expense and things like that. So our research is fungi and bacteria for my colleagues to do that. So effectively, bacteria like you and I we have fingerprints. Okay, we have DNA. You do DNA analysis, effectively it's your DNA fingerprint. Bacteria have the same thing. It's based on basically what I call biological barcode. So what you're going to see when we talk about, is it gram-positive or gram-negative? Is it a rod or a cocci or a spiral? Is it live in the air or does it live without the air? Is it an endospore former or is it not? Those are like yes/no questions. So you might have a skinny line, like a barcode, skinny line, dark line, skinny line, dark line. And whatever pattern that is somewhat of a unique pattern to very, very many bacteria. That's kind of what you do a little bit in the lab to use this sort of yes or no key to be able to analyze something. Is you do a series of tests. Does it have a certain enzyme? Does it not? Does it ferment to sugar? Does it not? And there's different sugars. There are typically sucrose, lactose, glucose that are involved. So those inclusions, what got me off on that, inclusions are accumulations of granules that don't do a whole lot. Maybe they're storage materials. Maybe they are sometimes little metallic particles in exotic bacteria. So we use them more for identification because they stain differently. There's a bacteria, when you sample your skin or the bottom of your shoe, which I think some of you probably have done, and start to plate that out, what you're going to see is there's a particular species that when you do a simple stain with methylene blue, kind of has interesting kind of an appearance. They're called metachromatic granules, and those are really inclusion bodies. You'll do endospore staining. It's a very interesting stain. Not a lot of things make endospores. That was the whole botulism tetanus number that we did the other day with Pasteur and all that stuff. Endospores are forever. And the interesting thing is, with endospores, we classify gram-positive rod bacteria. First classification, endospore or non-endospore. If it's an endospore form, it's a limited company. If it's not, it's another company. And that's how we begin to analyze those that are there. And yes, there are microcompartments, and most of this stuff is observed in archaea, which we really don't talk about. This is the other illustration why I didn't use it. Because that's the one gripe I have about this textbook and its PowerPoint sequence. They're putting a bunch of stuff in very small print that maybe you can, when you're looking at this electronically on your laptop, you can isolate and look at. It's very hard to teach with. But it's the same thing. These are little granules. This is a plasmid. This is the nucleoid or bacterial chromosome. It's all coiled up. Then you have the different layers that are there that we'll go over. You can see the flagella. These are little fimbriae. They're calling them pili here, but more likely they're fimbriae, these little bristles that use for attachment, et cetera, et cetera. And we'll start going over stuff. So if you want to read through that, we're going to see a summary of all of this. The first thing is how we classify. It's like anatomy and physiology. How do we know it's a lining cell? A lining cell, remember, it's either squamous or columnar or cuboid. Are these ringing a bell, these terms? See? You took that class. I taught you guys Okay? So it's all about classification. So I'm sitting here spouting off about gram-positive and gram-negative. Bacteria basically come in three shapes. First thing they're going to do, gram-positive, gram-negative. Okay, now we got that done. What flavor are they, so to speak, is that? And then, are they cocci, little spheres or circles? Are they rods? Another name for rods is a bacilli. That's the plural. Notice I use a small b because bacillus is a genus for one of those endospore forms that you utilize in the lab. Or are they a curved form? There's a variety of curved forms. And then there are spiral forms called spirilla. And there's other forms that look like a corkscrew, spirochetes. So basically we have cocci, rods, and then forms that are curved or spiral forms are sort of in one big group. And so those are things you look at in the lab. You look at these microscopically. This is sort of a crossover lab lecture kind of a group. So even though they function independently, you're going to see we're going to talk a little bit about biofilms. That sugar shell, particularly the ones that's kind of sticky, they form on surfaces, they form films that have sometimes single, many times multiple bacteria that collectively have impact. It's the kind of thing we would talk about for remediation with oil spills, would be something like that. One that we all have one carrying with us right now, dental tartar, is a biofilm. It's got many bacteria that adhere to the enamel surface of our teeth. The surface that's adjacent to the tooth, when it forms, is an anaerobic environment. It means there's oxygen, even though you're breathing all the time, doesn't get through. And anaerobic bacteria that cause cavities, they utilize that. Okay, the film weakens the enamel, the coating, and then the karyogenic cavity-forming bacteria. So when you get your teeth cleaned, or if you floss, or you use a tartar preventative thing like that, right? I don't want to delve into mouth care, but that's kind of the way it was. My mom was fastidious about that. Her sole goal before she died was to make sure she had all of her hair and all of her teeth. And she was able to achieve that. Lived to be 93. So we'd be going to a restaurant and she'd get up right after dinner and she says, and I said, is everything okay? She said, I gotta go floss. That was I'm keeping my teeth and I'm keeping my, she was the little old lady who always had the standing appointment Fridays at 10 o'clock at the beauty parlor to get her hair done. You have grandmas like that. Her last 15 years were in Rochester, New York. There was a foot of snow. She would drive. Nothing is stopping me from getting my hair done this morning. There's a foot of snow, Mom. Don't care. I have an appointment. Bruce. So all of those are things. So they communicate. We call them nanotubes, these channels that are open. Communication is like, hey, there's a tooth over there. Get ready. It's basically giving off chemicals that induce certain changes. They don't talk. Well, maybe they don't. Maybe they talk, but they don't listen. Thank you. One person gives me a smile. Partial. The rest of you. Okay. Very small. A micron. Again, maybe a tenth of a red cell, a tenth of a white cell. Certainly. Circular rods, a little bit longer. These other guys kind of go, pleomorphs are not quite those because they're a little more organized. Pleomorphs mean, and we'll look at one of them called, it's called it'll come to me, mycoplasma. And mycoplasma causes walking pneumonia, one of the things it causes. They change with the environment they're on. So they don't have a cell wall that's particularly rigid that's there. And so they're pleomorphs. They're caused by changes, nutritional, environmental conditions. So coccus or cocci is the plural. There are ones you'll see, Staphylococcus is very spherical. Sometimes they'll look like coffee beans. Sometimes they'll be a little more oval. And that's a characteristic in the lab that sometimes you can utilize. Bacilli are even more diverse. And again, notice we're using not the genera with this capital and the italicized letters, but bacillus is a generic term for rod. And they can be skinny, they can be fat, we call them coccobacilli, they can be chained together, they can have sort of funny looking ends, all of those types. There's a lot of variation. This one's very famous. There's an anecdote I share about it. It's called Legionella pneumophilia. Because it's a disease that periodically rears its ugly head that can be fatal, called Legionnaire's disease. I have it, I'll give you I gave the other guys the anecdote, but I can't really do it justice until we get to respiratory, so you can learn about it. That are there, respiratory diseases. But I was there. It's named because of the American Legion. We first noticed it, even though it was not what they thought it was originally, at the American Legion Convention in Philadelphia at the Bicentennial in 1976, summer of 76. And it happened to at the old Bellevue Stratford Hotel in the Rittenhouse Square section of Philadelphia. If you're familiar with Philadelphia, it's a neat district. Older, it's not the historic district. It's a few blocks away from that, but neat hotels, shops, restaurants, very charming, and good-sized student population. And that Bellevue Stratford is a little bit like our William Penn, that same genre, age, probably built immediately post-Civil War, let's say. And the Legionnaires all gathered in there, and it was people from World War I. They thought it was a recurrence of the dreaded Spanish flu. And remember, I told you my virology professor was the lead virologist for the Commonwealth. So he was tasked to see if it was. He couldn't find an answer. The reason was it was a bacteria, because everybody thought it was a virus. And it's a bacteria that grows in groundwater and dust and dirt and humidity. And these older buildings were grandfathered in with crawl spaces. They'd have the ventilation and the electricity and the hot and cold water lines. These old buildings that the service people go and crawl through. And if it leaked with the dust, it grew, and they all stayed in the same group of rooms and inhaled this repetitively, and about 15 of them died of what looked like flu-like symptoms. It's really very similar. I was there. We had our honor society meetings from school there. And the food, and I didn't have to pay for it, and the food was good. Let's look at what's important. Here are the curve shapes. Vibrio, very famous for being comma shaped like that. You heard of cholera as a dreaded disease. Vibrio cholera gets its name. Campior, we nickname it Campylobacter. Jejuni, it's kind of this sort of spiral shape, and there's a kind of food poisoning that comes from that. It's not terrible, but it's there. And then spirochetes. I drew it like a corkscrew. They look kind of like that. Okay, so that's an actual micrograph that's formed. So it's a spring. This is magnified 7,500 times. Two diseases that we encounter continually here with some degree of frequency. One a lot, Lyme disease caused by a spirochete. The other, syphilis, legendary disease caused by a spirochete. And then these filamentous shape, they actually reproduce at the tips. And what's interesting about them, it looks like spores. They're really not. But so filamentous bacteria have a big role, as well as endospore forming bacteria and making antibiotics. Like, for instance, that Z-Pak comes indirectly from a species of this, Streptomyces. They named the first of this group of antibiotics streptomycin, although it's not used very much today. But it was named for this. So arrangements vary. It's how they replicate. So if you have a bacteria that replicates in a plane, it's like one here and then the next one here in a single plane. Sometimes they go in multiple planes. Sometimes they're geometric. And we name them for that based on their appearance. Another thing, you look under the microscope. They can, E. coli, single. Okay. Diplococci, pairs. Bacterial meningitis, pairs. Okay. Tetrads aren't that important for us, at least clinically. Staphylococci, big clusters, because it replicates and they kind of continue to adhere from each other. So Staphylos represents a grape-like cluster of structures. Strepto means chains. So streptococci can either be chains or pairs, but it's very linear. And so when you hear about streptococcal pneumonia, which is the leading form of bacterial pneumonia, it's also called Diplococcus pneumonia because normally it's in pairs, and it's called pneumococcus, even though there are those three names for the same bacteria. And they're just showing you some, and you'll see a lot of this. Again, it's overlap left. This is called palisades because they adhere and create what looks like a hinge. They used to call this, because it was pejorative, they don't do it anymore, a Chinese letter pattern. Some of those environmental samples, we have these all over our skin and all over our environment. They're called diphtheroids. The most important one of these is Corynebacterium diphtheria, which is a dreaded disease. That's one of the 3 immunizations with tetanus, diphtheria, and pertussis, which is whooping cough. So diphtheroids look like it, but don't have it. And that's one of the ones where with stains, they form these metachromatic granules I was talking about with methylene blue. And moving on. So the flagella facilitates their movements. And our flagella in a sperm cell is just back and forth. These are different. They spin. And I was showing you a video. I'm not showing you, but just cooking up a video if I wanted to use it. Their flagella are like a helicopter. They basically twirl. And they can twirl in one direction or in the opposite direction, whether they move forward or away from something. Bacteria are simple. They're governed by what are called attractants or repellents. They're either going to move towards something, whether it's temperature, warmer or colder, whether it's light, if they're photosynthetic, whether it's air, if they're aerobic or anaerobic. Okay, even metal particles will exert magnetism will exert it, because some really interesting bacteria have magnetic like particles in them. Most of them move to chemicals. They're attracted by certain chemicals or they're repelled. So you have positive movement or negative towards or away. And that comes into this next nugget. So the filament, again, not microtubules, made of these repeat units of flagellin, a hook, and how it attaches or secures itself to the membrane is the basal body. So this is the basal body attaching it within the membrane proteins. This is the hook, which to me kind of gets it away from the remainder of the cell itself. And then you have the filament, which is the part, and you can see how this would spin. And you can see the micrograph, how we came to this conclusion with regard to it. We use it in the lab, and you'll probably look at prepared slides. I don't do the flagellar stain slides because the chemicals are kind of nasty, and you have to do them under the hood. So I'm kind of careful with that. I mean, maybe they made them better now. You can do it. So we characterize them on where the flagella is. That helps us identify. So, for instance, E. coli typically is monotrichous, has a single flagellum. It's got a bunch of different. Lopho is a small tub. That's what we have. Mono's pretty obvious. Trichus is like here. Amphi, to me, that's like ambidextrous. One on each side, one on each pole. Sometimes it's a tuft on each pole. And then peritrichus, it's surrounded. Those are swarming bacteria. You work with one of those in the lab called Proteus vulgaris. When you plate it out, it tends to have a very distinctive pattern how it grows on the plate. And this is what scientists use in every hospital. This is a series of things that they use to identify the bacteria. Because once you identify it, then you can determine what it's sensitive to, in other words, what antibiotic to use, and what would be the best one based on all things, allergies, side effects, etc. So, and they give you some illustrations that are okay. Here's a single polar. This is a tuft. I don't see the peritrichus here, but you can see those. And I think that other PowerPoint, the one from Nestor, which is Anderson's Microbiology, chapter 3. That's on week 1 for you. Probably has those illustrations. And here's the taxis. Taxis means movement. Okay. So the most common is certainly chemotaxis. And bacteria have limited energy resources. And this is what you were looking before. They do a thing called run, tumble, run. Let's say they're attracted. They'll move toward it when they expend their energy. Mostly they ferment, which doesn't make a lot of energy. Then they'll sort of tumble, sort of movement in no direction. Might be closer, might be farther away. Then we gather the injury and they'll move the energy and they'll start to move in some way towards it, then tumble and run. So you'll see some illustrations called run, tumble, run. Sometimes they just move randomly if there's no attractant or repellent in the area. So you see this, it depends on, and it has to do with how the flagella rotates. So a run, it moves in somewhat of a straight line. A tumble means it's probably spent its energy until it recoups enough to do another run. And it might end up moving farther away, be pushed by eddies and currents randomly. So this kind of, like here, if there's no attractant or repellent, there'll be motion, but it'll be, basically motion that's not necessarily going anywhere. And again, not all bacteria have the ability to move independently. A lot of them move by air and water currents. They're just in there, they move. And here, this is run, tumble, run. You're getting from point A to point B, but it's not like you and I walking to the door to leave the classroom. Unless maybe you're, overly tired. I was going to say had too much to drink, but we don't want to say that. Strike that from the record. I don't drink so much. Spirochetes, these spiral forms have what are called axial filaments. In between the cell wall and the cell itself, there's an interesting substance called periplasm we'll look at in gram-negative cells. And it's like if you were in scouts or you do camping and you tried to rub 2 sticks together. You typically what they used to do is they'd have like a string and you could rotate the stick this way. You know what I mean? To create movement back and forth. That's kind of like doing that. So it's like it's these filaments are twisting back and forth, and in doing so, this spiral structure moves. So they're called, and it's again something that you only see in these spirilla or spirochete forms, these axial filaments that are there. And then we have the other projections, the fimbriae, little bristles, like I said, ones that get in your sweater. These would be like gastrointestinal cells. And really, their little spikes are what attach to certain receptor sites on sensitive cells. You see, if you took a respiratory virus or a respiratory bacteria, so you're talking bacteria, its fimbriae, if you swallowed it, it wouldn't make any difference. Because in order for it to hurt you, it has to attach. Bacteria and viruses, too, are very specific onto the type of cell that they adhere to and interact with. So when you see certain things, depending on if your skin is intact and you get a virus that might hurt you, as long as you get it off your skin and your skin wasn't open or had a cut or an open wound or something, it wouldn't hurt you. I mean, I'm not recommending sticking your hand in a vat of E. coli, but it's a gastrointestinal virus. As long as you've washed your hands thoroughly, you're going to be fine. That's why you emphasize the hand washing part for all that, because you're not likely to touch your face and all that stuff. Pili are a little bit different. They're made of a protein called pilin, and rather than being, and it looks kind of like this, they saw that, and they typically are gram-negatives, look at that gram-negative. They saw a cell that had projection on it. And then later they saw in the same population this. And they thought that this was some type of sexual reproduction. The reality is that bacteria are asexual in nature. But this is an appendage called a pilus that was a hollow protein. And it's through that, when the cells connected here, that they could transfer genetic material, like I was talking about, from one to the other. They were calling it male and female, but the reality is that wasn't the case. So you had, and so you see, we're going to, when we get to the genetics unit, we're going to talk about the fertility, or F. pilus. And some authors just classify pili and fimbriae and use the terms almost interchangeably. But when we do the genetics unit, this is a big player in something called conjugation. As you can see, there are little tubes and things like that. And even here, as you see, within these little channels, even in the cell or in extensions of the cytoplasm, they kind of gobble up. Bacteria have a tendency to gobble up iron-rich substances. They thrive on that. And so when you get a bacterial infection, your liver sequesters iron. We try to limit the ability for the bacteria to use it, to grabs onto. So here you see this illustration all the time. Fimbriae are the little bristles, while the pili are much larger and longer, and they connect. And it doesn't have to be just one, you could be more than one interacting with another. The S layer is a protective layer that we don't really talk about that much, but bacteria, we've got to treat bacteria like living things, because they are. And they alter in an environment, and they are able to survive. That's the part we didn't understand when we used to use powerful antibiotics for no good reason, for long periods of time. People developed resistance, people developed side effects, people developed allergies. And this is the note about the slime layer and the capsule. Slime layers, loose, sticky, hold on to things. The capsule is very significant in disease. It's thicker, and macrophages, which is how we defend ourselves, and swallow up the bacteria and destroy it. Even if it swallows it, it can't destroy it, or it's too big to swallow. So capsules play a role. And so, I mean, this can be considerably thicker than is depicted here. And you'll do this stain in the lab, typically, if not for this bacteria, maybe for yeast, which have a distinctive capsule. And it kind of, you stain the exterior, and what doesn't stain is really where the bacteria in the capsule are. It's kind of like a little kid's light bright. Somebody here had a light bright. Terrible. Oh, God. You're not old, you're in college. So capsules are big players, problems, because white blood cells can't phagocytize them. Biofilms, the whole teeth thing, not just teeth. Catheters. For you nursing people, you're going to get sick and tired of hearing about catheters when you get into nursing classes. A major source of infection. Very hard to deal with regard to antibiotics. Okay, IUDs, if they're still used as intrauterine devices, and I don't know. Pacemakers, orthopedic implants notoriously pick up different kinds of staphylococcus and are very difficult to treat, because not every antibiotic can penetrate that. This is that nugget about the biofilm on the surface. You get all these little interesting tubes, and it's a complex interaction of multiple bacteria. Like I said, against, let's say if that's the tooth enamel, it creates an environment that facilitates cavities. And I just want to show you this, because this is how I left it. I told you about the envelope. Combination of cell wall, cytoplasmic membrane, and if present, the outer membrane. This is what we're going to start with, and probably spend really half the class on Monday. This is gram-positive. This is gram-negative. They're both bacteria. They're totally different. Inside, they're bacteria. The nucleoid, ribosomes. Here, the membranes are the same. This has a massive amount of peptidoglycan. This has a scant amount of peptidoglycan. This is a phospholipid bilayer. This is a phospholipid. It's this one in pink. That's the lipopolysaccharide layer. That's the odd one. And because it's membrane here and membrane here, it's tough to get through a membrane with a lot of medications and a lot of antibiotics. It's easier here because this is an airplane course. So we have to find out which one it is to determine how to go about treating it, what kind of problems it causes, and we're going to deal extensively with the intricacies of that structure at our next gathering on Monday. Thank you. Have a great weekend. Oh, my poor voice will never be